So we started regulation of amino acid biosynthesis, and somehow my laptop get hanged, and so I have to restart it, and then again we are starting with the same slide. Uh, uh, Here you can see that threonine hydratase is feedbackly inhibited by its end product isoleucine. Okay, so this is the rate limiting and committed step of isoleucine biosynthesis. And you can see this is just a good example of feedback inhibition via the product of same pathway. Okay, so now the another mode of inhibition or regulation we can see, another mode of regulation of amino acid biosynthetic pathway is known as concerted inhibition. This is actually also a type of allosteric inhibition in which one or more products inhibit the rate of biosynthesis of amino acid via different manner. Okay, for example, the best example of this is glutamine synthetase enzyme of E. coli. Here actually 6 products derived from glutamine. Okay, glutamine family actually involves for the synthesis of 6 products like AMP, CTP, adenosine monophosphate, cytidine triphosphate, tryptophan, histidine, carbamoyl phosphate, and glucosamine 6-phosphate. Okay, so these 6 products derived from glutamate, this serves as a negative feedback modulator of the same enzyme. So remember what it means, that concerted means the pathway is same. Here glutamate is converted into glutamine in presence of enzyme glutamine synthetase, but the same enzyme is inhibited by 6 byproducts. Okay, 6 byproducts: AMP, CTP, tryptophan, histidine, carbamyl phosphate, and glucosamine 6-phosphate. Here, one thing also you should remember that glycine and alanine separately inhibit this enzyme. So this enzyme contains a allosteric inhibition site where these molecules get bind and they inhibit the activity of this enzyme. And once the activity of glutamine synthetase get inhibited, the overall process of biosynthesis of all of these like glutamine, AMP, CTP, tryptophan, histidine, carbamyl phosphate, glucosamine 6-phosphate, it get inhibited. So this is the concerted mode of inhibition where only one enzyme is inhibited by several of byproducts of the pathway. Okay, and another is here one factor one should also remember is enzyme multiplicity. Enzyme multiplicity actually prevents one biosynthetic end product from shutting down key pathway when other products of same pathway are required. That means actually glutamine synthetase, as you see, it involves in the synthesis of glutamine only, but glutamine involves in the biosynthesis of so many products which are very vital and necessary for the life of an organism. So if this pathway get inhibited, then definitely the organism will die. Okay, so there are some bypass mechanism to prevent this, and this is known as enzyme multiplicity. That means there are some alternative pathways present. Okay, for example, you can see here 3 isozymes of aspartokinase is present in E. coli. Each responds differently as far as feedback inhibition and repression of enzyme synthesis. Threonine, lysine, methionine pathways independently controlled. Okay, so actually enzyme multiplicity is nothing but it is a it is a bypass mechanism. If one pathway gets shut down, it should not affect the overall process of the organism. So there are multiple isoenzymes present for that particular reaction, which can carry out and supply the products of demand. So this is just an example. You should remember this. And okay, Neha, you can ask question.
मैम अगर इनिबिशन रिक्वायर्ड है उस टाइम पे, अगर इनिबिशन रिक्वायर्ड है तभी वहाँ पे फीडबैक इनिबिशन या कॉन्सेंट्रेटेड इनिबिशन हो रहा है, तो अगर वो रिक्वायर्ड है तब हो रहा है तो फिर आइसो एंजाइम्स की जरूरत क्यों पड़ेगी जब वो प्रोसेस नीडेड ही नहीं है तो?
सी अगर बॉडी में आपका क्वेश्चन अच्छा है आपका, लेकिन अगर मैं आपको कहूँ आप इन प्रोडक्ट्स को देखिए, इन प्रोडक्ट्स को सीटीपी, एएमपी, ट्रिप्टोफैन, हिस्टीडीन, कार्बोमाइल फॉस्फेट एंड ग्लूकोसामिन 6 फॉस्फेट बन रहे हैं इससे। ओके, अब अगर मान लो आपका स्टमक फुल है, ओके, और स्टमक में आपका ये सब सामान की जरूरत नहीं है, तो ये स्टमक के ग्लूटामिन सिंथेटेज को इनिबिट कर देंगे, समझ में आ रहा है। लेकिन अगर बॉडी में किसी और जगह पर एएमपी और सीटीपी की रिक्वायरमेंट है, ओके, किसी और प्रोसेस में ट्रिप्टोफैन की रिक्वायरमेंट है और ये एंजाइम ओवरऑल पूरी बॉडी में इनिबिट हो जाएगा, तो बताइए कैसे फिजियोलॉजिकल प्रोसेसेस आगे चलेंगे। ठीक है, तो इसीलिए आइसोएंजाइम, जैसे इसका बेस्ट एग्जांपल मैं अगर आपको दूं, लैक्टेट डिहाइड्रोजनेज। ओके, आपको पता है मसल में अलग आइसोएंजाइम प्रेजेंट है, हार्ट में अलग और लिवर में अलग। ओके, कारण ये है कि अगर लिवर के पास सफिशिएंट लैक्टेट है तो वहां का लैक्टेट डिहाइड्रोजनेज इनिबिट भी हो जाए, बट हमारा मसल का काम करना चाहिए, क्योंकि मसल तो हमें दिन भर यूज करनी है। ओके, राइट। ऐसे सिमिलरली हार्ट हार्ट तो कंटिन्यूसली चलता रहता है। अगर ऐसे ही लैक्टेट जो सर्कुलेशन में है वो अगर सभी मतलब फीडबैक इनिबिशन अगर कर रहा है और सभी लैक्टेट डिहाइड्रोजनेज को इनिबिट कर देगा, क्योंकि अल्टीमेटली वो ब्लड के थ्रू सभी को कर सकता है ना। अगर सभी इनिबिट हो गए तो हमारे हार्ट का क्या होगा, है ना। हार्ट को तो कंटिन्यूअसली एनर्जी चाहिए, कंटिन्यूअसली पाथवे उसके चलते रहना चाहिए, तो इसलिए आइसो एंजाइम का वहां पर रोल होता है। जैसे अगर आप ये एक एग्जांपल हुआ, दूसरा एग्जांपल आप हेक्सोकाइनेज और ग्लूकोकाइनेज का भी ले सकते हैं। लिवर का जो ग्लूकोकाइनेज है और जो ओवरऑल यूनिवर्सल हेक्सोकाइनेज है, इन दोनों की एफिनिटी टुवर्ड्स ग्लूकोज अलग अलग है ना। इस तरीके से अगर हम कहते हैं कि हेक्सोकाइनेज का फीडबैक इनिबिशन जो इनिबिटर है, जरूरी नहीं है ग्लूकोकाइनेज को भी उसी लेवल उसी एक्सटेंट तक इनिबिट करे, क्योंकि अलग अलग ऑर्गन के एंजाइम्स का अलग अलग लेवल ऑफ डिग्री ऑफ इनिबिशन अगर मैं कहूँ तो वो अलग अलग होता है। तो आई होप आपकी क्वेरी सॉल्व हुई होगी।
यस मैम।
ओके, तो यहाँ पर इसीलिए आइसो एंजाइम का फंडा चलता है। ओके, तो एंजाइम मल्टीप्लिसिटी जस्ट यहाँ पर एक फैक्ट था तो मैंने बता दिया। नेक्स्ट, कमिंग टू द नेक्स्ट टॉपिक, दैट इज आवर सीक्वेंशियल फीडबैक इनिबिशन। See, first you should know about what is the difference between concerted and sequential. Okay, in concerted you can see there is only a single reaction, and after this single committed reaction multiple byproducts are synthesized during different pathways. Okay, multiple byproducts synthesized through different pathways, but all of the bioproducts, if they get accumulated in higher amount, they inhibit the key reaction. Okay, all of them inhibit a key enzyme only, but in sequential this is not the case. Actually in sequential, step by step inhibition happens. Okay, like aspartate is first converted into, just it is just this pathway is for your understanding and just for explaining you how it works exactly in body. Aspartate is first converted into aspartyl beta phosphate. Okay, and there are three isoenzymes present, A1, A2, A3. Okay, and aspartate beta semialdehyde is converted into homoserine, and two isoenzyme B1 and B2 is present here. Homoserine is converted into methionine and threonine. We have seen this pathway earlier in biosynthetic reaction, and this threonine is further converted into alpha ketobutyrate and then to isoleucine. So this is just a basic pathway where if isoleucine is accumulated in the body, it inhibit the key enzyme of A1 only, okay, not the A3. Okay, so this is actually something different, and this A3 isoenzyme is inhibited by threonine. Okay, and threonine can also inhibit simultaneously this B2 enzyme and this homoserine to threonine converting enzyme. Okay. Similarly, methionine can inhibit this homoserine step. Lysine can inhibit this aspartate beta semialdehyde step, and plus this can also inhibit the A1. So this is the sequential manner. In this slide, if I can sum up this slide, in this slide, the synthesis of lysine, methionine, and threonine and one isoenzyme for requires one isoenzyme for conversion of aspartyl beta phosphate, which is allosterically inhibited by two different modulators, lysine and isoleucine. Two different modulators. These are entirely different, lysine and isoleucine. These two inhibits the same enzyme. Okay, isoenzymes of the same reaction. And the sequence from aspartate to isoleucine undergoes multiple overlapping negative feedback inhibition. You can see there are multiple overlapping negative feedback inhibition. Cross sign indicates always negative feedback inhibition. So, for instance, isoleucine inhibits the conversion of threonine to alpha ketobutyrate. Okay, and like this step, alpha ketobutyrate and threonine inhibit to alpha ketobutyrate. This threonine basically inhibit to alpha ketobutyrate, and then thereby it also inhibits its own formation. Okay, threonine also inhibits its own formation. So this is this steps occur in 3 points from homoserine to aspartyl beta semialdehyde and aspartate to, and this aspartate beta semialdehyde to homoserine, and this homoserine to methionine, and methionine to isoleucine. So I hope you can understand this sequential feedback inhibition. So in amino acid biosynthesis, we have learned today about feedback inhibition, concerted feedback inhibition, and sequential feedback inhibition. We have simultaneously um, learned about the difference between sequential and concerted feedback inhibition. If you have any kind of doubt, you can ask, or otherwise we can move to the other part. Okay.